Imagine a world ravaged by infections. A world where every minor injury, every small cut or scrape could spiral into a life-threatening situation. A world where a simple cut could lead to death. Infections were rampant, and medical science was often helpless against them. This was the harsh reality before the advent of antibiotics. Doctors and nurses did their best, but without effective treatments, many patients succumbed to infections. In 1928, a Scottish scientist named Alexander Fleming made a discovery that would change the course of history. His work would revolutionize medicine and save countless lives. It was a simple accident, a moment of carelessness that led to an extraordinary breakthrough. Fleming left a petri dish of bacteria uncovered, an oversight that would prove to be serendipitous. When he returned, he found it contaminated with mold. But instead of discarding it, he observed something unusual. This was no ordinary mold. It was producing a substance that seemed to kill the surrounding bacteria. The mold was actively destroying the bacteria, creating clear zones where the bacteria could not grow. This mold was Penicillium, and the substance it produced was penicillin, the world's first antibiotic. It was a groundbreaking discovery. Fleming's accidental discovery marked the beginning of a new era in medicine. His work laid the foundation for the development of antibiotics. It was the dawn of the antibiotic age, an age where we could finally fight back against bacterial infections that had plagued humanity for centuries. This discovery has saved millions of lives and continues to be a cornerstone of modern medicine. Before penicillin, even minor infections could be deadly. A simple scratch could lead to sepsis. Childbirth was a life-or-death struggle for both mother and child. Infectious diseases like pneumonia, tuberculosis, and typhoid fever were major killers. Doctors had few tools to fight these infections. There were no antibiotics. Treatments were often ineffective and sometimes dangerous. The discovery of penicillin was a beacon of hope in this dark age of medicine. Alexander Fleming was a brilliant but often absent-minded scientist. In 1928, he was working at St. Mary's Hospital in London. He was studying Staphylococcus bacteria, a common cause of infections. Fleming went on vacation, leaving his lab somewhat messy. Upon his return, he noticed something peculiar in one of his petri dishes. A mold had contaminated the dish, but the area around the mold was clear. The bacteria had not grown there. Fleming realized the mold must be producing something that killed the bacteria. He identified the mold as Penicillium notatum. He named the antibacterial substance it produced penicillin. Fleming's initial discovery was just the first step. In 1928, Alexander Fleming noticed that a mold called Penicillium notatum had killed bacteria in a petri dish. This accidental discovery was groundbreaking, but it was only the beginning of a long journey. Penicillin was difficult to produce in large quantities. The early attempts to extract and produce penicillin were met with numerous challenges. The mold was finicky, and the yields were incredibly low. It was also unstable and hard to purify. The compound would break down quickly, making it nearly impossible to use in a clinical setting. Scientists had to find a way to stabilize and concentrate it. It wasn't until the late 1930s that a team of scientists at Oxford University, led by Howard Florey and Ernst Chain, were able to isolate and purify penicillin in usable quantities. Their work was meticulous and required innovative techniques to extract the antibiotic effectively. They proved its effectiveness in treating bacterial infections in animals. This was a significant milestone, as it demonstrated that penicillin could be a powerful weapon against infections that were previously untreatable. In 1941, the first human patient was treated with penicillin. This marked a pivotal moment in medical history, as it transitioned from experimental to practical use. A policeman named Albert Alexander was dying from a severe bacterial infection. The penicillin worked miraculously. His condition improved dramatically. This case provided undeniable proof of penicillin's potential to save lives. However, the supply of penicillin ran out before he could make a full recovery. The scarcity of the drug was a major issue, highlighting the need for mass production. He tragically died a few days later. This heartbreaking outcome underscored the urgency of producing penicillin on a larger scale. Despite this setback, the potential of penicillin was clear. The scientific community was galvanized, and efforts to mass-produce penicillin ramped up. This led to the development of methods that eventually made penicillin widely available, revolutionizing medicine and saving countless lives. The wartime wonder drug. World War II provided a powerful impetus for the mass production of penicillin. Before the war, penicillin was a promising but scarce antibiotic, discovered by Alexander Fleming in 1928. However, it was not until the war that its true potential was realized. Infections were a major killer on the battlefields. Soldiers often succumbed to wounds that became infected, leading to severe complications and death. The need for an effective treatment was urgent. The Allied governments recognized the potential of penicillin to save lives. They saw it as a critical tool to reduce the number of fatalities and improve the recovery rates of injured soldiers. They invested heavily in its development and production. This investment included funding for research, building new facilities, and training scientists and workers to produce the drug on a large scale. Pharmaceutical companies in the United States and Britain worked tirelessly to scale up production. They faced numerous challenges, including finding the right strains of mold and developing efficient production techniques. New methods of fermentation and purification were developed. These innovations allowed for the mass production of penicillin, making it more accessible to those in need. By D-Day in 1944, enough penicillin was available to treat all Allied soldiers wounded in the invasion of Normandy. This marked a significant milestone in the war effort. Penicillin became known as the wonder drug that saved countless lives during the war. Its effectiveness in treating infections was unparalleled, and it quickly became a standard treatment in military and civilian hospitals. It transformed the way battlefield injuries and infections were treated. The success of penicillin during World War II laid the foundation for the development of other antibiotics, revolutionizing medicine and saving millions of lives in the years to come. The dawn of a new era. After the war, penicillin became widely available to the public. Its impact was immediate and profound. Diseases that were once death sentences became treatable. The death rate from bacterial infections plummeted. Life expectancy increased dramatically. Penicillin became the cornerstone of modern medicine. It paved the way for the development of other antibiotics. The discovery and development of penicillin marked a turning point in human history. It ushered in a new era of medicine, the antibiotic age. The enduring legacy of penicillin, a cornerstone of modern medicine. Penicillin's legacy extends far beyond its initial discovery. It revolutionized the way we treat bacterial infections. It led to the development of countless other antibiotics. These drugs have saved millions of lives and continue to be essential tools in modern medicine. Penicillin and its successors have made it possible to perform complex surgeries, organ transplants, and chemotherapy, procedures that would be too risky without effective antibiotics to prevent and treat infections. The challenge of antibiotic resistance. The widespread use of antibiotics has led to the emergence of antibiotic resistance. Bacteria, through the process of natural selection, are evolving to become resistant to the drugs designed to kill them. This is a serious threat to global health. Infections that were once easily treatable with antibiotics are becoming harder and, in some cases, impossible to cure. The World Health Organization has declared antibiotic resistance one of the biggest threats to global health, food security, and development today. The future of antibiotics. seeking new solutions. The fight against antibiotic resistance is a constant battle. Scientists are working tirelessly to develop new antibiotics and alternative treatments for bacterial infections. Strategies include developing new drugs that target different mechanisms in bacteria, using combinations of antibiotics to overcome resistance, and exploring new approaches like phage therapy, which uses viruses to kill bacteria. Public awareness and responsible antibiotic use are also crucial in combating resistance. It's essential to use antibiotics only when necessary and to complete the full course of treatment as prescribed by a doctor. Conclusion: Penicillin's enduring impact. Alexander Fleming's accidental discovery of penicillin was a pivotal moment in human history. It ushered in the antibiotic age, transforming medicine and saving countless lives. While the challenge of antibiotic resistance reminds us that the fight against infectious diseases is ongoing, penicillin's legacy continues to inspire new discoveries and innovations in the quest for a healthier future. The story of penicillin is a testament to the power of observation, the importance of scientific research, and the enduring impact that one accidental discovery can have on the world. If you found this video interesting, don't forget to like, subscribe, and share. And check out more fascinating discoveries that change the world.